Okay, we'll move on now to the next talk, and we'll age a little bit with our Down syndrome population. Today I want to talk about biomarkers in Alzheimer's disease in Down syndrome. So what I'm going to describe today will be an ongoing study that we've had going on for the last 6 years. I want to provide a little bit more detail on what we're researching here at the Waisman Center. So just as a brief outline, why are we interested in Alzheimer's disease? I think there was just a brief discussion of that in Down syndrome. Want to give a little bit of the background of Alzheimer's disease, as well as biomarkers we're going to be looking for. Detail some of the findings that we've reported in our research so far in Down syndrome, as well as kind of the latest update on what we're finding in Alzheimer's disease in general with our imaging study, and then go to what's new in our research happening here at the Waisman Center. So adults with Down syndrome are at a very high risk for developing Alzheimer's disease, with most individuals, once they get over 40, are showing some of the pathology with Alzheimer's disease, and I'll explain exactly what I mean by amyloid deposits in a bit. So our goal in this research is to find some of these markers of Alzheimer's disease in adults with Down syndrome, and track these over the course of life. Look at their changes in cognition, imaging changes that we're seeing as far as using medical imaging technologies, and biochemistry, and how these things will change with their functioning over time. And really, we're hoping that this will help guide us in understanding better the transition to Alzheimer's disease in Down syndrome, exactly what's going on in the brain at that time. Our goal is to understand it not only for the Down syndrome population, but hopefully to translate that into the general population, understanding what are the targets that we're looking for in Alzheimer's disease, really help us identify potential targets for treatment. And so Alzheimer's disease in Down syndrome. First of all, in the general population, we have a prevalence of about 5% at age 65, and then it gets older. Roughly a third of the adults, as we get into our 80s, will have Alzheimer's type of dementia. So really, age is the #1 risk factor. The older we get, the more likely we are. Now, if we look at the Down syndrome population, though, we have roughly the same proportions, however, at a much younger age. Individuals in the 40s are developing roughly 10%. Then as we get older into our 50s, now we have about 1/3% of the population. So it's about 3 decades earlier that it is onset. And then as we get into our 60s, we have a very high risk for developing that. So let me give you just a brief background on Alzheimer's disease. I'm sure everybody in here knows someone with Alzheimer's disease. You have a very good understanding of what it is. So it's a type of dementia where we see a deterioration of cognitive function that really keeps a person from not only living independently, but really functioning on a day-to-day basis. And Alzheimer's disease is a type of dementia. It's the most common type of dementia. 70% of dementia is Alzheimer's disease. And there we have symptoms, difficulties in language, in memory, behavior, cognitive skills, executive function, just things that really help us make good decisions as we function throughout our days. So of course, Alzheimer's disease is a big public policy issue. Over 5 million Americans and 100,000 Wisconsinites have Alzheimer's disease. It is rising on our ladder as far as the cause of death. And of course, our population is growing older. We're going to see the number of cases of Alzheimer's disease increasing. So we're very fortunate here at the University of Wisconsin to have some incredibly valuable resources. We have an NIH-funded Alzheimer's Disease Research Center across the street in the hospital. I work very closely with many of those investigators. I'm part of that. And of course, our Waisman Center here, where we can identify populations to kind of really use the tools that we've developed with our ADRC to study this particular group of people. So why is Down syndrome seem to have a higher incidence of Alzheimer's disease, or why is our trajectory altered in this case? Well, we know Down syndrome is trisomy 21. We have 3 chromosome 21s. And if you look at the chart I have on the right, it just is a cartoon of all of the chromosomes. And everywhere you see a little colored section in those chromosomes, that indicates a gene that is implicated in increased risk for Alzheimer's disease. And if we look at chromosome 21, let me see if I can get my mouse going here, we see an increased, or we see a marked area on the chromosome. And that is a gene that's responsible for this APP, amyloid precursor protein, for the production as well as maintenance or regulation of that particular protein. And I'll talk a little bit more about that in a moment. But the running hypothesis is you have an extra chromosome, therefore there might be overproduction or at least dysregulation of this amyloid precursor protein, which could be leading to some of the pathology that we see in Alzheimer's disease, which I'll give just a brief overview of right here. So when Dr. Alzheimer first looked at individuals in the early 1900s in examining their brains post-mortem, he saw 2 kind of signature presences in the brain, and these were referred to as plaques and tangles. So it's pathology that I'll highlight a little bit here in the next slide, but in essence we are seeing cell death to the neurons, and we have a pattern illustrated as the disease progresses. And what I want to show is right in this area is where it starts early. That's the hippocampus in the brain. It is used, very involved in memory, long-term memory, as far as memory formation and memory retrieval. And then as the disease advances, we see it spread throughout various regions of the brain in general, afflicting almost the entire brain. So first of all, let's consider those plaques that I mentioned. So this little cartoon just illustrates the surface of maybe the cells within the brain, and tethered to their surface you can see this APP. That's the amyloid precursor protein that I was just showing you that's implicated with chromosome 21. And under normal conditions, we have some enzymes there that can cleave this off. Now, the APP is part of normal physiology. It belongs there. And in certain pathways, we have these enzymes that can snip it off the cell surface, and it's soluble, and it floats away and is washed out of the brain. Under different situations, though, we have an enzyme here. I showed the purple one there. It's called beta secretase. And that can cleave that APP off the cell surface in a slightly different place. And now you're left with a different little fragment there, illustrated in yellow. And in this one, that little peptide fragment is now sticky, and it doesn't clear out of the brain as easily. Rather, it sticks together, and it forms what are referred to as oligomers, and the oligomers then bind to these fibrils, and all of a sudden you get this large plaque, amyloid plaque, that can coat the surface of the neurons, coat the different cells there, and eventually have toxic effects in the brain. So this is one of those signatures of diseased brain that Alzheimer first noted. And the other are these tangles. And this is another protein in the brain. And these tangles, or tau, is a protein that forms kind of the scaffolding on the neurons. It keeps the structure together. And they can become unbound, and they can form these tangles. And these tangles then can deposit in the cells. And these too can cause cell death. So once again, we have plaques and tangles in the brains of individuals with Alzheimer's disease. And these can potentially become biomarkers for identifying AD. So a brief explanation, a biomarker is a biological measure that is an indicator of Alzheimer's disease, or something that indicates disease progression. And it is very useful in medicine to find biomarkers for different diseases. It helps us track progression, and we also have something to focus on if we want to look at therapies that will slow down or halt the progression of disease. So with Alzheimer's disease, and this is all research going on over at the ADRC, as well as nationwide. We have different types of biomarkers for Alzheimer's disease. They can be in the cerebrospinal fluid. You can see little fragments of those proteins, either the amyloid or the tau, as well as blood-based measures. There can be genetic measures that are biomarkers, and also imaging measures. And that's what we're focusing on here very heavily in our research, looking at signatures in the brain that can indicate something with regards to the progression of Alzheimer's disease. And then there is behavioral and cognitive testing too. So I want to give you some introduction as well as some of the results we found in brain imaging in individuals with Down syndrome, and trying to identify those biomarkers for Alzheimer's disease. And we use two different imaging modalities. The first, MRI. I would assume everyone in here has probably had an MRI before. It's simply a magnet that submits radio frequency signals, and from that we can get these exquisite looking images, very high resolution, and in the brain they're very useful. We can see gray matter and white matter and all the different structures in the brain. And what it primarily defines, we call MRI typically, it provides information on structure. That is, the different areas of the brain. And if we go over to PET imaging, which is positron emission tomography, now we have a modality that we call function. That is, the way this works is we can label different drugs or molecules or whatever you can think of that would be of interest in a disease process and inject that into the body. And we give this in very small amounts, that is tracer amounts. So even though I'm saying we inject drugs into the bodies, they're in such small amounts, they don't have any type of drug effect. But they go to the same place that these drugs will. So for example, we can label a particular drug of interest. One of them might be FDG, fluorodeoxyglucose, or glucose. Glucose is your very basic sugar, and that's how the brain derives almost all of its energy. So if we can inject some of this FDG into a person and image them with PET, we can see the areas of the brain that are being used in metabolizing glucose. So if I let you look at this for a moment, on the top I show what we would call healthy control, and then on the bottom is an Alzheimer's subject. I hope you're kind of comparing the two and trying to identify what's the difference in the Alzheimer's disease subject. First of all, you see a lot of glucose throughout the uptake, but there are a couple regions that show reduced metabolism. And those being the arrows, the parietal cortex shown in this particular slice, as well as right up here. So now we have this imaging modality that gives us a biomarker of Alzheimer's disease. However, this is a person with advanced disease state, and we are always looking for biomarkers that present earlier and earlier indications. Another PET drug that we've been studying is this PIB, that stands for Pittsburgh Compound B, and we collaborate with the investigators at the University of Pittsburgh. And we've been doing a great deal of research in this area. So now if we look at these PET images of the brain, first I'll direct you down to the bottom row here. And this would be the pattern we would see of PIB binding in a healthy control, which is what HC stands for. And you see just the bluish color in the middle of the brain. And that is what we refer to as off-target binding. That's in white matter. We aren't particularly interested about it there. However, if we control it with the subject with Alzheimer's disease, now you can see the red areas of high uptake in some of the regions of the brain, similar to where we saw the decrease in the sugar utilization. So once again, here in this parietal area, and then this precuneus area too. So this PET tracer has been used now for about 10 years, and we're getting fascinating information in the general population and imaging with it. So what we find, up here, is individuals with Alzheimer's disease, if you image them, you're going to see high uptake of this tracer. And that is, it marks these amyloid plaques. And by definition, if you have Alzheimer's disease, you have amyloid plaques. So no surprises there. However, if we look at the healthy control, as well as this intermediate state called MCI, or mild cognitive impairment, now we're seeing some people with reduced uptake, or amyloid, and some with elevated. So even though people might have it, they still function properly. So we're very intrigued by what this is telling us. And I'll explain a little bit more about that in some slides. And what's happened since now, I came and spoke at this meeting several years ago, and since that time, we only had availability to that PET tracer that looked at the plaques in the brains. Well, now within the last couple of years, there are tracers available that label the tangles too. So research is just starting looking at tangles. And here is a comparison of some of the studies that we have done with the Alzheimer's researchers. So in a healthy control up on top here, you can see the amyloid image, and there really isn't any areas of increased uptake, just kind of the yellow and green showed there. And then also, here is this tau tracer. It's called THK5117. We really need to come up with better names for many of these. But that doesn't happen until drug companies get hold of them and you get marketing people involved. These are all so far designed at universities, and we aren't as creative in that sense. But we don't see the high uptake in this healthy control. You might see some in the middle of the brain. Once again, that's off-target binding. We aren't really sure yet what to make of it. But if we go down now with this individual with Alzheimer's disease, you can see the high uptake in amyloid shown in this top row. And if you go down to the bottom row, now we can see increase in this tau protein that we're seeing too. And the area we're most interested, I showed before, is the hippocampus, which is right in there. And we see areas where we have increased uptake. So we're thinking this might track a little bit better with cognitive decline, because we know that the amyloid, people can bear a burden in their brain and still function quite well. So we're very interested now in tracking studies. We've done roughly 30 of these studies so far here, and next year we'll have hundreds, and probably in a couple of years we'll have thousands. So a very interesting story is now unfolding with regards to these different biomarkers in the brain. And it helps us derive conceptual models. That is how we think it's going to happen. And what has really been kind of characterized so far is that the first curve, that black and green one, shows amyloid. And it just suggests that that starts accumulating in the brain relatively early. And the brain, like I said, can bear a lot of it before we start to suffer any type of cognitive deficits. However, the other markers tend to come at lower levels later. And here, this is an indicator of tau, this light blue one, that will be tracking over the time ahead. Okay. So now I'd like to go back to our Down syndrome population and talk about our ongoing study that for the last 6 years we have been researching here at the Waisman Center. So we're bringing in individuals with Down syndrome that are 30 years and older, and they are all, none of them are showing signs of dementia at all. That is, they aren't having any cognitive problems as far as remembering things. And we're doing scanning exams on them using this amyloid tracer I was talking about. And then we're also doing a series of cognitive measures. So for the first day, they'll come in and do a series of tests. We have these different kind of games they play to measure cognitive functions. And then the second day, they come in and we do our scans. And here's an example of what we're finding with our amyloid images in Down syndrome. And that is areas of increased uptake you can see right here in kind of the middle of the brain, as well as in the cortex. And people, in other individuals, don't have that pattern. Once again, in the middle, we aren't as concerned. We like to focus on more the outside of the brain where the cortex is. So here are some results with the University of Pittsburgh. Between both of our sites, we're in the mid-70s as far as the number of people that we have scanned. And these are different brain regions. And really, all I want to show you, if I pick one in particular, the striatum's kind of the middle of the brain. And this is arranged according to age. And our subjects, the youngest are coming in at 30 years, and our oldest are in their 50s. And what we're finding is that there are quite a few individuals that we have that are showing increased binding or increased amyloid, and they're starting out at a relatively young age, so the mid-30s. In the healthy population in general, we don't see binding this early in individuals. And if we kind of look at the different patterns we see, of the 72 subjects we found, 20 of them are showing increased uptake of this amyloid, whereas 50 of them aren't. So we are just now undergoing the longitudinal scans. That is, we're coming and we're going to track them at various time points, come in 2.5 years later, and we can track to see if there's any change in their brain scans as well as their performance on these tests. So as I said, only people that are non-demented are coming in for our study. And we want to now compare this to see if we can find any behavioral or cognitive biomarkers that would suggest the presence of this amyloid in the brain. So we've divided them into individuals who showed increased binding of the amyloid and ones that did not, and I'm calling them positive and negative, or PIB positive and PIB negative. And we're starting to compare them with some of the behavioral studies they have done. So one of them, for example, is the Vineland Adaptive Behavior Scales, and it just tells their independent living skills, their ability to communicate and socialize. And we haven't found any difference between them. That is, subjects with increased amyloid really aren't showing any difference there. If we look at behavioral measures, such as aggressiveness, paranoia, depression, there we are not seeing any differences between the groups, so everything seems to be fine there. However, once we focus on memory functions and we really interrogate memory a little bit more, now something starts to come out. So here's an example of what we refer to as the cat and dog Stroop test. So the person will flash up these pictures, and all they have to do is name the animal. So for this, it would be cat, dog, dog, cat. And then we tax the system a little bit more, and they have to identify the object, but suppress the urge to say it, and instead switch it to the other name. So in this instance, you would say dog, cat, cat, dog. And once we do that, now we see a little bit of a separation between the groups. That is, the individuals with the positive amyloid didn't perform as well on the test as those that did not have it. And here's an example of a working memory task where the psychologist will tap out a pattern on the block, and then the subject has to follow that pattern. And in the forward direction, no problem. But if we make it a little bit more difficult and have them reverse the pattern that they saw, now we start to see a difference emerge there too. So we are seeing alterations just slight changes in cognitive function in these individuals. So if I'll go back to the general population in what we're learning, more studies are starting to come out. We're starting to track individuals throughout the course of life, as well as if some will transition from normal functioning into mild cognitive impairment or Alzheimer's disease. And this is the model we're getting. And finding out that with the amyloid, it's a very slow process. Here, this is 30 years. We might start to see accumulation. And it climbs, and eventually a person will develop dementia. But even this isn't fixed yet. We still don't completely understand the role of this amyloid. It is not for certain that somebody will develop Alzheimer's disease. We're still looking that closely. We know that many people have it in their brain and can function quite well. So this is what we're hoping now to derive a scale like this for Down syndrome and see what the trajectory is for this population. And once we define it, that will help guide us as far as therapies, possibly participating in drug trials. There are drugs now being developed to break up these amyloid plaques in the brain. And then here's another finding in the general population. I realize it's kind of a busy slide, but if you just look at this, it illustrates a couple things, and that is the accumulation is very slow. This shows the amount of increase in amyloid per year as people come back in for scans. And in general, we're looking at just a couple % of increase. So it's a slow process. But also, look at the spread in this data. It's very, very broad as far as where a person is. So still, we have to understand these biomarkers. How sensitive are they going to be to changes? How much variability do we see in them? Okay. So with that, I'd just like to finish up talking about the new research we have going on in Down syndrome. So this was a study that I have just described with the University of Pittsburgh. We were following individuals with Down syndrome, looking at the behavioral measures as well as the amyloid in the brain. It's been funded since 2010. We've found very interesting results. And recently, we were awarded a relatively large-scale grant to expand this from the National Institute of Health. We brought in Banner Institute at the University of Arizona as well as Cambridge in the United Kingdom. And we're referring to this study as NIAD, Neurodegeneration and Aging of Down Syndrome. We're increasing our numbers. We're getting larger numbers. Because you see all that variability in the data, we need a lot of individuals to really track it and try to understand what's going on. And we're going to add more biomarkers, not just our imaging of the amyloid, but we're going to start imaging tau as well as the glucose. And then we want to look at blood as well as CSF. Just look at all of these different biomarkers that are being developed for the general population too, and really trying to characterize these in their progression as we go on. So I'll give some conclusions here. As Maria's just suggested, there's been some amazing advances in healthcare, and people are living longer with Down syndrome. And accompanying that, then you're going to see the old age type of diseases afflicting individuals with Down syndrome too, namely Alzheimer's disease. In our scan, we found about 30% of the subjects showed this presence of amyloid. However, they could still function very well, even though this was present in their brain. But we were able to see slight changes in cognition. So now we're looking forward in the future, getting more of these biomarkers, really trying to characterize it, and hopefully get involved in clinical trials with drugs to examine the effectiveness, hopefully treat Alzheimer's disease and advance it not only for the Down syndrome population, but for the general population too. So with that, I would like to thank you for your attention, and also I can't express how thankful we are for the people who participate in this research. We have the families will travel here and sometimes have to stay overnight. We can't say thank you enough to all of the people who have made this work possible. And